Good evening, children. Welcome to another practical assignment today. Before we begin, I'd like to show you how your practical file is to be divided into 5 segments. According to your syllabus, the first point is about scientific techniques. Second is physiology, which we have completed under 4 practicals. Third is morphology that we'll start today. Then fourth is cytology. And the fifth is spotting. So children, when you are making your file, here your second, like suppose we have done. So it will be shown like this. This is section 2, physiology. And under this we have done all 4 practicals. Now you can see the ordering could be in any number, but they should be compiled together at one place. I hope you understand. Then similarly, when we are starting today with our section 3, morphology, another paper like this you'll write section 3 and below morphology. So today's practical is on a fungus. We call it as bread mold or rhizopus, and we will be learning how to prepare a temporary slide of the same. As the name tells, children, this fungus is called as bread mold, right? Because it usually grows on bread, but at the same time it can be grown on other articles like syrups, jellies, leather, etc. So if we keep a moist bread and cover it by a bell jar for some days, what we see is a whitish cottony outgrowth on this substance or the substratum. In this case it is bread. Sooner this whitish cottony growth starts turning black because of the asexual structures that are found inside a bulb-shaped structure called as sporangium. The spores that are formed, they are black in color, so gradually the white color becomes black. Alright, so there are 3 types of structures seen. There are root-like structures, what we call rhizoids. They penetrate the substratum to obtain the nutrients. The sporangiophores, they are arising just at the same point but in opposite direction. So if rhizoids are penetrating into the bread, then sporangiophores are just opposite aerial, it is the direction, and erect these are, which finally end up in bulb-like structures called sporangium. I'll show you by another diagram here. This is the bulb-shaped structure called as sporangium. Now each thread-like structure, right, it is called as a hypha, and that is cenocytic. Cenocytic means there are many nuclei. So gradually what happens, there is a partition laid down called columella, right, and all nuclei, they are shifted on the other side. That means on this side there will be no nucleus left, and slowly the cytoplasm will divide around each segment of nucleus and form a spore. So the same spore, it is nothing but a broken piece of a nucleus and the cytoplasm that has broken around the nucleus, right? So here ultimately the black spores, they are released in the atmosphere, and wherever they fall will be another growth of the fungal material. So this is what you're going to learn how to prepare it, and I'll be giving you the same by a file like this the previous year. So let us see how you begin. It will be practical number 5. Today's date, that is 1st of August. The object, clearly, that is to prepare temporary slide of bread mold, rhizopus, after culturing it in laboratory. Stain the slide with cotton blue, mount in lactophenol, and observe under a compound microscope. The next heading is apparatus. A piece of or a slice of bread, bell jar, cotton blue, lactophenol, glass slide, cover slip, pointed needle, and compound microscope. Procedure. Keep a slice of bread in moist chamber for a few days till it gets covered by a black cottony growth on it. Pick up a few hyphae from the bread by using a pointed needle and transfer it on a glass slide. Third point, stain the fungus with cotton blue and mount in lactophenol. Fourth, observe the structure of bread mold under the microscope and write its details. The next heading is observation. Here you will write the points again. This is a slide of Rhizopus, commonly known as bread mold or black mold or pin mold, right? So it will be or black mold or pin mold. Second, mycelium is cenocytic. The term cenocytic actually means a multinucleate structure in which the nucleus, yes, has divided, but the cytokinesis has not taken place. That means imagine if a cell has divided, the nucleus, suppose it is like this, one nucleus, and it divides in such a way that many nuclei are formed, but the cytoplasm has not divided along with it. So such a cell will be called as cenocytic. Alright, so this is the same condition of the hypha or the thread over here, or the mycelium is a tuft of hyphae or a group of hypha. Mycelium is cenocytic, profusely branched, and appears as white cottony mass. Three types of hyphae. See, three types of hyphae mean there are rhizoids that are penetrating into the bread to absorb nutrition. There are transverse structures, the second type of hypha called as stolon. And the third types are the erect sporangiophores just opposite the tuft of rhizoids or the group of rhizoids. So, three types of hyphae, the stolons, rhizoids and sporangiophores are recognized in the mycelium. What is mycelium, children? Suppose this is the fungus in groups, right? And the same is growing on a piece of bread. So one thread will be called as hypha, right? The plural of hypha is called as hyphae, right? So hypha is a single thread-like structure, but in this knot or this structure which is a group-like structure we call as mycelium. The plural of the same, this is called as mycelia. So remember how you have to understand hypha. When we are talking of a single thread-like structure of the fungus, right? Plural hyphae, but the group or cluster or tuft of the same called as mycelium, and the plural is mycelia. So you're talking of the same hyphae which are cenocytic, that is multinucleate without the division of cytoplasm. Three types of hyphae, stolons, rhizoids and sporangiophores are recognized in the mycelium. Next point, stolons grow horizontally on the substratum and develop tufts of rhizoids at point of contact. Next, sporangiophores are erect, aerial and unbranched hyphae which grow upwards in tufts from the stolons exactly opposite to rhizoids, and they bear terminal sporangia. So you saw here how the sporangiophores, they are arising in tufts or clusters just opposite the rhizoids, and their terminal end they are having the bulb shaped or rounded sporangium. Now sporangium is a singular term, the plural is what you call as sporangia, right? So sporangia is a plural term. Next point, the sporangium has a columella in the center, and the space between columella and wall of sporangium is packed with aplanospores. So what happens is there's a partition between the wall of sporangium here, right, and the hypha over here. So this is in the center, that's a columella present, right? So between columella and the wall of sporangium, they are filled black colored spores, right? And the same we are calling as aplanospores. Aplanospores mean that they are not motile. Motile means they cannot move, and they are asexual spores. So I'll give the meaning. Aplanospores are non-motile asexual spores. That means they are being used in asexual reproduction and they cannot move on their own. So how do you think they will move? Only by the effects of wind currents or by air. Alright, so this is how the term aplanospores they mean over here. Next point, the aplanospores are globose, that means rounded, multinucleate, non-motile structures. After dehiscence, what is dehiscing, children? The sporangium. As you can see, it cannot bear the pressure after certain time, and the wall of sporangium bursts open, liberating the spores in the air. So after dehiscence or the breaking, the remnants of the sporangial wall can be seen as a frill at the base of columella, right? And the result, the above experiment demonstrates preparation of temporary slide of rhizopus. So that's all for the working today, children. All this we will conduct only when the school reopens. But just now, because we are maintaining the regular assignment with our writing part, it is important that you write it, right? I'll be sending you the diagram that you have to draw. You will have to draw two diagrams. One is the simple working culture of rhizopus in laboratory, and the other is the structure of rhizopus. So I'm going to send you today's first. I understand there was some error in the uploading of files, but now it has been rectified, children. So the same assignment you will be handing over or submitting by 5th of August. So I'm giving you a good amount of days where you can complete the same. Any queries, children, feel free to ask, because now we'll be more in touch with our WhatsApp group. So you can ultimately get chance to call me or message me or ask any doubts. Thank you.